welcome to today's video lesson. So far, everything we've learned about replication, transcription, and translation, we've been talking about in eukaryotes. So there are some things that differ in transcription and translation in prokaryotes, and we're about to discuss those briefly now. So the first thing is that, you should know this already, that prokaryotes don't have a nucleus. So their DNA is located in the cytoplasm, where eukaryotes, their DNA is in the nucleus. That's nothing new. Okay? Prokaryotes have circular chromosomes. So it's because they don't have linear chromosomes, they don't have ends of chromosomes, and so therefore they don't have the telomerase problem, where DNA polymerase can't end to nothing. Because prokaryotes chromosomes are circular, they don't need telomerase, because there's no ends of chromosomes. And eukaryotes have linear chromosomes, which have ends, and that's why we need telomerase. Two other differences. The first one you're not going to understand, because we're actually going to get that to the week before Christmas. Prokaryotes have naked DNA. There's nothing on their DNA. While the DNA of eukaryotes is wrapped around histone proteins, and we're going to talk more about that the week before Christmas, so don't worry about that right now. The last difference is the major difference, is that prokaryotes have no introns in their DNA, so they don't need spliceosome. They don't cut out the introns. Whereas eukaryotes have both introns and exons, and the exons get cut out. I hope you heard Janet. So just to summarize, because I forget what I said, prokaryotes don't have introns, so spliceosome doesn't need to cut anything out. They don't even have spliceosome, where eukaryotes have introns and extrons, so the introns need to be cut out. Another thing that's important about at least translation in prokaryotes, or the production of protein, is that since there's no nucleus, once transcription happens, immediately after the mRNA comes out, the ribosome hops onto it, okay, and does translation. There's no time gap. Remember, in eukaryotes, the mRNA is made in the nucleus, and then it has to travel from the nucleus into the cytoplasm to the ribosome. In prokaryotes, that's not true, okay? As soon as the DNA is made into mRNA, the ribosome hops on it immediately. There's no mRNA editing, there's no need for a GTP cap, there's no need for a poly A tail. As soon as the mRNA is transcribed, it is immediately translated into protein. So once again, this is really sort of the main difference, again, in the time and physical separation between the processes of transcription and translation. So in prokaryotes, transcription and translation happen upon immediately. As soon as the mRNA is transcribed, it's translated. So you can see that here. Here's the mRNA that's transcribed, and immediately upon its transcription, it's translated into protein. Whereas in the eukaryotic cells, the DNA needs to be processed before it can be translated. So we have our mRNA, and the mRNA needs to be spliced, and we have to add our GTP cap and the poly A tail, and then it has to travel from the nucleus into the cytoplasm. So there's a time gap in eukaryotes that's not present in prokaryotes. Okay, so we're switching gears a little bit now, and we're entering a part of the course, or part of this unit, that's about how genes are controlled, so how genes are turned on and off. We're first going to look at this process in prokaryotes, and then we're going to look at this process in eukaryotes. In eukaryotes, it's much, much more complicated. So first, we're going to look at how prokaryotes, or bacteria, as we see here, control their gene expression. On the AP exam, they're going to use the word gene expression a lot, okay? And if you're doing gene expression or gene regulation, okay, regulation means you can turn a gene off, and if you turn a gene off, it makes no protein. Or, the reverse of turning a gene off is you can turn a gene on, and then it makes protein. So the process, okay, of turning a gene on and off is called either gene expression or gene regulation. Another term they could also use, if they say a gene is expressed, that means that it's turned on and it's making a protein. If they say a gene is not expressed, that means it's turned off and it's not making a protein. So, just to confirm that you've written this down, gene expression or gene regulation is the process by which genes are turned on and off in organisms. We are now going to look at this process in prokaryotes, or at least the beginnings of this process. So, a gene, just to review one more time, is a segment of DNA. On the test, when it asked for a gene, you all gave me the RNA. So a gene is a segment of DNA, okay, that codes for a protein. Okay? Now we can turn genes on and genes off. Okay? If we don't need that protein at the current point, we want to turn the gene off. Okay? Or if we have enough of it, we don't need it anymore. If we want it, then we need to turn the gene on. So this lesson, okay, the next 2 days, really 3 days actually, or actually when we get to eukaryotes, maybe the rest of this unit almost, okay, is on how genes are turned on and how genes are turned off. So this slide I meant to take out because we're going to skip. We're going to skip this slide, cross it out. So by regulating their gene expression, okay, that allows bacteria to respond to changes in their environment quickly. So for example, if they have enough of something, okay, or enough of a protein, not something, of a protein, they want to stop its production. They don't need to make any more of it. Okay, and the reason is that if they make something they have enough of, okay, they're wasting the energy to make these things that they don't need, they have enough of. And they're going to do this by stopping the transcription and translation of the genes that code for these things. Similarly, okay, bacteria may find a new food source that they need to break down to use as energy. Okay, and remember, they need energy so that they can grow and have ATP and all that loveliness. So they're going to want to start the synthesis, okay, or make the proteins or enzymes that are able to break down this food source so that they can use it for energy. Okay. If you remember way back when, hi Ralph, there's Ralph. Okay, I forgot about Ralph. We haven't used Ralph in a while. So if you remember way back when, we learned about this concept called feedback inhibition. Okay, this was like in early September, where, okay, if you have a process, so all these molecules are, you're taking this original molecule here and turning it into tryptophan, okay, all these molecules are like sort of precursor molecules in the process, okay, and they're regulated, or the process is done by these enzymes. If you remember when we were learning about feedback inhibition, when you make enough of the final product, it goes back and allosterically inhibits the first enzyme that made it. And if you remember, allosteric inhibition is when something, in this case tryptophan, binds to the enzyme and changes its shape, or changes the shape of the active site, which many of you didn't write on the last test. Then it can't function anymore. And that makes sense because you don't want to make any more tryptophan if you have enough of it. So it goes back, when there's enough of it, it goes back and inhibits the enzyme that makes it. Okay, but think about it. The process of even making these enzymes, enzyme 1, enzyme 2, enzyme 3, is wasteful if you don't need them. So why even try to go and inhibit the enzymes when you can just inhibit the genes or turn off the genes that make the enzymes in the first place if you don't need that? And that is actually what bacteria do. Bacteria. Sorry, that's not the right plural. So we're going to see how bacteria can both turn on their genes when they need them and turn them off when they don't need them. And the purpose of turning, or the benefit of being able to turn on and off genes, is that you only have the proteins made when you need them. So like I said, instead of having tryptophan go back and inhibit the enzymes, okay, that make itself, it's going to go back and stop the production of the productions of the enzymes in the first place, so that we don't even need to make them. Okay. We're now going to look at how that's done in bacteria. How do we block transcription, or block the expression of genes in bacteria? Alright, so one more time. Again, we are going to talk about how genes regulate their transcription. And they do this by turning genes on and turning genes off. The next sort of like week or so, okay, is all about how this is done in prokaryotes, okay, and then how all this is done in eukaryotes. So for some reason, bacteria cells group genes together that are all involved in one process in something called an operon. Okay, now I know you do not have this picture, so what I would recommend doing is that you pause the video now and draw the picture below. Because I'm going to talk about the parts of the picture. In class, we're supposed to do this lesson in class. In class, I was going to draw it with you. But now we're not in class. So, you should draw it. And we're going to talk about the parts of an operon. So, again, like I said, an operon are genes that are grouped together that function in the same sort of pathway. So, in this below is the trip operon, which we're going to discuss. And these genes here, TRPE, TRPD, TRPC, TRPB, TRPA, they code for the enzymes that are necessary to make tryptophan. Okay, tryptophan is an amino acid, by the way. But again, these are the genes of the operon. Okay, and they are all these proteins or enzymes. Okay, same thing. Okay, are needed. TRPE, TRPD, TRPC, TRPB, TRPA are needed. Okay, to make tryptophan. There are also other parts of the operator that we need to look at. The first part here, this part here, is the promoter. Okay, and the promoter is where RNA polymerase binds. And remember, and this we saw this in transcription when we were learning it last week. RNA polymerase is the protein that turns the DNA into mRNA, so it binds to the promoter. Okay, there's another site that bacteria have called an operator. Okay, and the operator, okay, is where the repressor protein binds. So the repressor protein would bind here, and we're going to see the role of the repressor protein in a second. The last spot, which is actually not written in the text, is this spot here. This is called the regulatory gene, or TRPR, and the regulatory gene, right here, codes for the repressor protein. So it has the code to make the repressor protein, and it's actually always on. Okay, so that's the repressor protein gene. So the question is now, how, if we want to turn this right now, this gene would be transcribed, because you have RNA polymerase here, and it can go along and turn this DNA here into mRNA. So the question is, what can we do to stop these genes from being transcribed? Or basically turn gene expression off? And the answer is in the repressor protein. So when the repressor protein is active, so right now here it's inactive, okay? When the repressor protein is active, something can bind to it to make it active. When it's active, it binds to the operator, okay? And when the repressor protein is bound to the operator, it blocks RNA polymerase. And then since RNA polymerase is blocked from transcribing the gene won't be made into mRNA, and then the mRNA won't be, and then the mRNA won't be made into protein. So if we look at this slide, this is the last slide in this lesson, so we can take as much as we can time on it. This is the regulatory gene, okay, which makes the repressor protein. And then here the repressor protein is inactive. But if the repressor protein is activated, okay, the repressor protein will then bind to the operator and block RNA polymerase. Then transcription can't happen and the gene will be off. In the next lessons that follow, the next couple of days, we're going to be looking at two different operons, the TRP operon and the LAC operon, and how they're different and how they're the same from each other.